My dear students, I am Shafi, your biology teacher. I hope all you are safe at your home. I know that you are happy to be in the high school. In high school, basic science divides into three: physics, chemistry and biology. Now I am dealing with biology. I think you have an idea about biology. Biology means it's about living things. Example: plants and animals and other organisms. In your biology, the first chapter is the mystery, the life mystery in little chambers. That means it's about living things, or we are discussing about organisms. In this chapter, the important point is the little chambers. Little chambers means cell. I think you have an idea about the cell. You are already learned from your previous classes. Cell means it's a simplest, that means small particles. In your previous classes, you have observed some different cell structure in your science lab. In this chapter, we are learning about cell. Commonly, the cell we can't see with our naked eye. I think you have an idea about that. We want to observe a cell with the help of a microscope. So, first of all, we want to discuss a structure of a microscope. In this chapter, we want to discuss the structure of a microscope. The structure means the different parts of a microscope. I think you have an idea about microscope. You are already listened in your science lab. Maybe a chance you are observing some slides through microscope. Now we want to discuss the structure of a microscope. The structure of a microscope means that we are discussing the different parts of microscope. The first part of the microscope is eyepiece, and the second one, knobs. There are two knobs out there, and the third one, objective lens, and fourth one, stage and clip. Then a condenser, finally a mirror. There are commonly 6 main parts in microscope. Just we want to clarify the functions of these parts. The first one is eyepiece. Simply we can understand what is the need of eyepiece. We want to observe the object through this eyepiece. And the second one is knobs. These knobs are using for adjusting the length or the distance between lens and object. We are using the knobs for adjusting the objective lens from the object. And the another part is objective lens. Objective lens are different in some different microscope due to the power of that lens. If we want to observe a very small particle, we want to use a high power lens. So, objective lens is used for observing the smallest particle very clearly. Then another one, stage and clip. The stage and clip is used for observing. We are observing an object. We are just put into the stage, and we are using two clips for the accurate position of that object, for clearly observing that simple particles. Then a condenser is there. The condenser is used for observing the smallest particle. That means the intensity of light is maintaining this condenser. Intensity of light means if you are reflecting, commonly we use the microscope with the help of sunlight, and we are using the sunlight. The mirror is used for reflecting the light. That means we are using the mirror for reflecting the light, and the reflection passes through the condenser, and this condenser is used for maintaining the intensity of that light. That intensity, the intensity is helpful to observing the picture. That means the smallest particles very clearly. These are the different parts of microscope. All of you listen the class, because we are observing an object using the microscope. We have an idea about these different parts, because if we want to observe a smallest particle, for example, if you are observing a small plant, first of all we have an idea about these different parts, because we want to clearly observe the picture. How to adjust the knobs, then how to arrange the slide in the stage with using clips. That means with using clips, and how to increase the intensity of light with using mirror. Then how to adjust the distance with using these knobs, and we are observing through these eyepiece. I think I have an idea about this microscope. You should observe the picture and just remind the different parts names, and you just understand these parts is used for what. The microscopes are different types. If we want to observe a smallest particle, commonly in your science lab you are observing a compound microscope. A compound microscope means we are using lenses more than one. A simple microscope is used for observing the particles comparatively large. We are observing through a compound microscope. That means if we want to observe a small particles if you are using compound microscope. The compound microscope means in this compound microscope have more than one lens. Simple microscope have only one lens. You just remind there are two microscopes are there, simple microscopes and compound microscope. Simple microscope means a single lens, a single objective lens is used for compound simple microscope, and the compound microscope means this compound microscope is used more than one lens. Okay, I hope you have an idea about this microscope. Then listen, my dear students. Next, we want to discuss how to observe a cell through the microscope. How to observe a cell through microscope. I think you are very interested to conducting an experiment in science. All students are very interested for about that. But now we are unlucky. You just listen carefully what I say. If we want to conduct a simple experiment, that means how to observe a plant cell through a microscope. Not only plant cell, we want to observe animal cell and other small organisms, unicellular organisms like amoeba and paramecium, like that. But now the availability of the simple cell is very easy to avail. Very easy available is plant cell. That's why we are taking a plant cell. You can just take a leaf of a plant or the small plant's stem. You can use whatever you like.
All of you listen. Here let us perform an activity to observe a plant cell. First of all, take a small portion of plant stem. Cut the thin cross section of the stem with the help of a blade. Put the material to be observed in the petri dish containing water to prevent hydration. Petri dish means it's like a watch glass and it contains some water to prevent the dehydration of the thinnest cross section of that plant part. Take thin cross section of the material to be observed. With a brush, shift the section to a watch glass containing water. Put the thinnest and complete sections to a watch glass containing stain. The stain is used for coloring and observing the plant cell with very clearly and understanding the different parts of that plant cell. Then after, add 1 or 2 drops of glycerin to the slide to prevent drying up of the material. Maybe a chance to drying up of this material. We want to prevent that drying by adding glycerin. And wash the stained section in clean water and place it in the glycerin on the slide. The glycerin is used for preventing drying. Then after, place a cover glass and cover the material using a needle in such a way that air bubbles do not pass. We want to prevent the air bubbles for neatly observing the plant section through microscope. So we are using needles for preventing air bubbles. Then wipe out the excess glycerin using tissue paper for neatness of that slide. Then after the preparation of that slide, we are observing the slide through microscope, and finally we can see a very neat and clean plant cell. That means we want to see a smallest particles in our world. We can't see with our naked eye, so we are using microscope for observing micro particles. So I hope all have an idea about this, and this activity, that means this preparation and observation of material through the microscope. Okay, then please carefully observe the different parts of microscope, and all of you read and well study these parts. Okay, thank you.